Chapter 1, Section 2, Characteristics of Cell Membranes. Cell membranes are composed primarily of lipids and proteins. The lipid component consists of phospholipids, cholesterol, and glycolipids, and is responsible for the high permeability of cell membranes to lipid-soluble substances such as carbon dioxide, oxygen, fatty acids, and steroid hormones. The lipid component of cell membranes is also responsible for the low permeability of cell membranes to water-soluble substances such as ions, glucose, and amino acids. The protein component of the membrane consists of transporters, enzymes, hormone receptors, cell surface antigens, and ion and water channels. Phospholipid component of cell membranes. Phospholipids consist of a phosphorylated glycerol backbone, the head, and two fatty acid tails, as shown in Figure 1-2. The glycerol backbone is hydrophilic, that is, water-soluble, and the fatty acid tails are hydrophobic, that is, water-insoluble. Thus, phospholipid molecules have both hydrophilic and hydrophobic properties and are called amphipathic. Figure 1-2, shown on page 4, illustrates the amphipathic nature of these molecules. The polar portions are soluble in water, and the long fatty acid tails are soluble in oil, giving them the appearance of a bilayer shown in Part B of this figure. At an oil-water interface, see Figure 1-2a, molecules of phospholipids form a monolayer and orient themselves so that the glycerol backbone dissolves in the water phase and the fatty acid tails dissolve in the oil phase. In cell membranes, see Figure 1-2b, phospholipids orient so that the lipid-soluble fatty acid tails face each other and the water-soluble glycerol heads point away from each other, dissolving in the aqueous solutions of the ICF or ECF. This orientation creates a lipid bilayer. Proteins in cell membranes may either be integral or peripheral, depending on whether they span the membrane or whether they are present on only one side. This distribution of proteins in a phospholipid bilayer is illustrated in the fluid mosaic model shown in Figure 1-3. This is Figure 1-3 from page 5 in the book, illustrating the fluid mosaic model. Integral membrane proteins are embedded in and anchored to the cell membrane by hydrophobic interactions. To remove an integral protein from the cell membrane, its attachments to the lipid bilayer must be disrupted, for example by detergents. Some integral proteins are transmembrane proteins, meaning they span the lipid bilayer one or more times. Thus, transmembrane proteins are in contact with both the ECF and ICF. Examples of transmembrane integral proteins are ligand-binding receptors, for example, hormones or neurotransmitters, transport proteins, for example, sodium-potassium ATPase, pores, ion channels, cell adhesion molecules, and GTP-binding proteins, or G proteins. Other integral proteins are embedded in the membrane but do not span it. Peripheral membrane proteins are not embedded in the membrane and are not covalently bound to cell membrane components. They are loosely attached to either the intracellular or extracellular side of the membrane by electrostatic interactions, for example, with integral proteins, and can be removed with mild treatments that disrupt ionic or hydrogen bonds. One example of a peripheral membrane protein is ankyrin, which anchors the cytoskeleton of red blood cells to an integral membrane transport protein, the chloride bicarbonate exchanger, also called the band 3 protein. This concludes Chapter 1, Section 2.